And this is about chromosomal theory of inheritance, and this was proposed by Sutton and Bovary in the year 1902. And they observed that chromosomes from two parents, that is a maternal and paternal, male and female, come together in the zygote as a result of the fusion of two gametes, male and female gametes, and again separate out during meiosis at the time of formation of gametes. Okay, and chromosomes are filamentous bodies present in the nucleus and seen only during cell division. And these are the basic information about chromosomes, and what is the number of chromosomes in zygote and gametes also. Such informations are given here. Gametes have half the number of chromosomes or n number of chromosomes and also said to be haploid. On the other hand, zygote is diploid or said to be 2n or it has double the number of chromosomes when compared to chromosome numbers present in the gametes. See, for example, in human beings, sperm has 23 chromosomes and ovum has 23 chromosomes. When these gametes fuse together to form a zygote, diploid condition is restored. That is 2n number of chromosomes. So the individuals have 46 chromosomes. These observations proved that there is a remarkable similarity between the behavior of Mendelian factors or genes during inheritance and that of chromosomes during meiosis. So this led Sutton and Bovary to propose this theory, chromosomal theory of inheritance. Okay, we shall pass on to the next one. This is the salient features of chromosomal theory of inheritance. So same information only what we have discussed already. So the somatic or body cells of an organism which are derived by the repeated division of zygote have two identical sets of chromosomes. That is, they are diploid. So somatic or body cells are diploid. Out of these, one set of chromosomes, that is, out of 46 chromosomes, 23 chromosomes are received from the mother, that is, maternal chromosomes, and the other set is from the father, paternal chromosomes. Two chromosomes of one type, that is, one type it refers to these chromosomes carry genes that control the same set of characters. So this is called homologous pair. So homologous pair of chromosomes refers to chromosomes carrying genes that control the same set of characters. And humans have 23 pairs of chromosomes. The chromosomes of homologous pair separate out during meiosis at the time of gamete formation. The behavior of chromosomes during meiosis indicates that Mendelian factors or genes are located linearly on the chromosomes. Okay, I hope you understand. Linearly, it refers to they are located one below the other on the chromosome. This is called linear fashion. So genes are arranged or located linearly on the chromosomes. So with progress in molecular biology, it is now known that a chromosome is made up of a molecule of DNA and specific sets of segments of DNA are the genes. Otherwise, we can say that genes are the segments of DNA. Genes, that is equal to segments of DNA, and segments of DNA, it is equal to chromosome. So we shall move on to the next one. So this is about linkage and crossing over. So Bateson and Punnett performed a dihybrid cross with two breeding varieties of sweet pea, Lathyrus sativus. And when they performed this experiment, dihybrid cross in this pea, a kind of sweet pea, they observed that instead of 9:3:3:1 ratio, which usually occurs in dihybrid cross, they got the ratio 7:1:1:7. So it means that the characters controlled by the two genes chosen for the experiment do not follow the principle of independent assortment as postulated by Mendel. You must have understood what is independent assortment. It says that genes for the two different features are passed down into the offspring independently. But it did not happen in this experiment performed by these two scientists. So instead, they tend to be inherited together, not independently. So chromosomes tend to be inherited together or linked together. Chromosomes in the sense genes. So thus, genes present on the same chromosome tend to be inherited together and are said to be linked. This phenomenon is called linkage. So, linkage is the phenomenon. It refers to genes which are arranged on the chromosomes close together. They inherit together and said to be linked. So, we shall move on to the next slide. So, this is about linkage and crossing over. All the genes present on the same pair of chromosomes and with a tendency to be inherited together forms a linkage group. Okay, linkage group, it refers to the genes present on the same pair of chromosomes and with a tendency to be inherited together. It forms a linkage group. In the experiment conducted by Bateson and Punnett, some recombinant type of individuals were also produced. Some new recombinants, which were not present, the traits were not present in the parent, so which are called recombinant. They are produced by another phenomenon called crossing over. So, crossing over, we must have learnt it in our earlier classes. It is the physical exchange of parts of the non-sister chromatids of the chromosomes of a homologous pair. Exchange of genetic material between the parts of the non-sister chromatids of the chromosomes of a homologous pair. So, crossing over occurs during prophase 1 of meiosis 1 at the time of gamete formation. The point where crossing over occurs is called chiasma and plural is chiasmata. So, linked genes get separated from each other by crossing over. Linked genes get separated from each other by crossing over. Because of linkage and crossing over, a heterozygous individual can produce 4 types of gametes. So, this is about linkage and crossing over, and this is the illustration that shows crossing over and recombination. So, this is a homologous pair of chromosome, and this is another one. These chromatids are called sister chromatids. Okay, so here crossing over occurs. So, homologous chromosomes, they come closer together so that crossing over can occur. So, here exchange of genetic material occurs. So, this is colored with shaded with a different color so that you can understand. This small blue shaded part, it is exchanged for this red shaded part. So, you can understand here. So, these are the recombinant chromatids. Okay, so this is what happens during crossing over. So, we shall move on to next one. So, this is about chromosomes and sex determination. Sex of the unborn individuals is determined in different ways in different kinds of organisms. We can easily understand, we can determine also the sex of the individuals by some methods. In some diploid organisms, specific chromosomes have a role in sex determination. So, we can understand it is the chromosomes by which we can determine the sex of the individual. So, such chromosomes are called sex chromosomes, and the rest of the chromosomes of a set are called autosomes. So, if sex chromosomes are morphologically similar, that is XX, like in female ovum, that is in ovum or female gamete. So, this condition is called homogametic because both are same, both the genes are same, or allelic pair, both are same. Such individuals produce only one kind of gametes containing X. So, they produce XX. And for example, all eggs of human female contain an X chromosome and autosomes. So, human female is termed as homogametic. So, this is the other sex determination occurs. Before that, this is about the chromosomal condition in males. So, sex chromosomes in males are morphologically dissimilar. It is XY, but in females it is XX. Such individuals produce 2 types of gametes, one containing X, the other gamete containing Y. So, they are called heterogametic. For example, human male produces 2 kinds of sperms, X-bearing sperm and Y-bearing sperm. When the human egg is fertilized by an X-bearing sperm, a girl is born, and if human egg is fertilized by a sperm having Y chromosome, a boy is born. So, XX girl, XY boy. Whether the unborn will be a male or female is purely a matter of chance, and no parent can be blamed for the sex of the progeny or the offspring.